Hello everyone, I'm Shen from Bio 110. Today I'm excited to share a topic that's both fundamental to biology and closely linked to our lab experience: enzymes, molecular engines catalyzing life. Through this video, I will connect what we learned in Bio 110 lectures with the lattice experiment we conducted, and also explore how enzymes impact our daily lives. Let's dive in. First, let's outline what we'll cover in this video. We'll start with a review of core enzyme concepts from our lectures. Then, we'll focus on the lattice catalytic activity experiment, its principle, materials, procedures, and experimental design. Next, we'll explore the real-world applications and biological significance of enzymes. Finally, I will share my acknowledgements. The experiment will be the centerpiece of today's talk, as it perfectly illustrates this theoretical knowledge we gained. Let's begin with the enzyme core background. Enzymes are biological catalysts. They speed up chemical reactions by lowering activation energy without being consumed in the process. This is a key property that makes them indispensable in living organisms. Enzymes also have three critical characteristics: high efficiency, meaning they can accelerate reactions millions of times faster than uncatalyzed processes; specificity, as each enzyme binds to a specific substrate due to its unique active site; and environmental dependence. Factors like temperature and pH can significantly affect their activity by altering their three-dimensional structure. The mechanism behind enzyme action is the induced fit model. Now we come to talk about activation energy comparison in detail. Here is the progress of the reaction. Every chemical reaction between molecules involves bond breaking and bond forming. The initial energy needed to start a chemical reaction is called activation energy, which is EA in this graph. In catalysis, enzymes or other catalysts can lower the EA barrier, thus speeding up specific reactions. We've also mentioned induced fit model when discussing the mechanism behind enzyme action. Let's demonstrate it now. When a substrate approaches the enzyme's active site, the enzyme undergoes a conformational change to better fit the substrate, forming an enzyme-substrate complex that facilitates the reaction. Beyond the lecture content, let's expand a bit. Enzymes can be classified into different types based on their functions. For example, lactase belongs to hydrolases, which catalyze hydrolysis reactions. Additionally, some enzymes require non-protein cofactors or coenzymes, such as metal ions or vitamins, to exert their catalytic activity. Now let's move to the cool part, our lattice experiment. The main objective of this experiment was to extract lactase from commercial tablets, determine its catalytic activity at 37 Celsius degree, the optimal temperature for human enzymes, and verify the enzyme's specificity and efficiency. But here's a challenge. Lactose and its hydrolytic products, which are glucose and galactose, don't absorb visible light, so we cannot directly measure the reaction with a spectrophotometer. To solve this, we used ONPG, O-nitrophenol beta-D galactoferrinoside, as a synthetic substrate. ONPG has a similar structure to lactose, with a beta-glycosidic bond that lactase can specifically hydrolyze. When lactase acts on OMPG, it breaks it down into galactose and O-nitrophenol. O-nitrophenol is a yellow-colored compound that absorbs light at 450 nanometers. By measuring the absorbance at 450 nanometers at different time points, we can indirectly quantify the amount of O-nitrophenol produced, which reflects the catalytic rate of lactase. This method leverages the Beer-Lambert law to link absorbance to concentration. To conduct this experiment, we used a set of specific materials and reagents. For reagents, we had OMPG as the substrate, PBS buffer to maintain a stable pH environment, and sodium carbonate as the top solution. Adding it increases the pH, inactivating lactase and terminating the reaction. For equipment, we used commercial lactase tablets as the enzyme source, a mortar and pestle to grind the lactase tablets, a dry bus incubator to maintain the 37 Celsius degree reaction temperature, a vortex to mix the solutions better, a microplate reader to measure absorbance, a centrifuge to separate the enzyme extract from solid particles, and various pipettes for precise liquid transfer. The experiment consisted of three main phases. First, lactase extraction. We ground one lactase tablet into a fine powder using a mortar and pestle. Then the powder was transferred to a tube and 10 ml of PBS buffer was added to create a suspension. After vortexing for 1 minute to maximize extraction, we transferred 1 ml of the suspension to each of the two microcentrifuge tubes and centrifuged them at 10,000 rpm for 1 minute. The supernatant we collected was the lactase extract. This step separated the enzyme from insoluble impurities. Next, the reaction setup. We prepared 2 control groups and 4 experimental groups with 3 replicates each. Control A had no enzyme to verify that OMPG wouldn't hydrolyze spontaneously. Control B had no substrate to rule out background color from the enzyme itself. According to data in the table, 14 tubes were labeled and prepared with fixed amount of PBS and OMPG. For each tube, fixed amount of lactase extract was added using P20 pipette. Then the tube was vortexed for 2 seconds and put into the 37 Celsius degree dry bus, with the timer being set at 30 seconds, 60 seconds, 90 seconds, and 120 seconds respectively. The tube was taken out of the dry bus and 500 microliter of stop solution was immediately added when the time was up at each predetermined time point. Finally, absorbance measurement. We transferred 200 microliter of each reaction solution to a 96 well plate and used the microplate reader to measure the absorbance at 450 nanometers. This step gave us quantitative data to analyze the enzyme's catalytic activity. A key strength of this experiment is its rigorous design. Let's break down the variable control. The independent variable was the reaction time, 30 seconds, 60 seconds, 90 seconds, and 120 seconds. The dependent variable was the absorbance at 450 nanometer, which correlates with O-nitrophenol production. We controlled several other variables to ensure the experiment's validity. Temperature was kept constant at 37 Celsius degree. pH was stabilized with PBS buffer. Enzyme and substrate concentrations were standardized by precise pipetting, and the total reaction volume was unified across all tubes. The experiment's design also emphasized scientific rigor. By including blank controls, control A and B, we eliminated interference from non-enzymatic factors. For example, we confirmed that the yellow color only came from enzyme-catalyzed OMPG hydrolysis, not spontaneous reactions or the enzyme itself. Having three replicates per experimental group reduce random error and also make our results more reliable. The time gradient design allowed us to observe the time dependence of enzyme catalysis, which is a key kinetic characteristic of enzymatic reactions. However, enzymes are not just lab tools, they are essential to life and have countless real-world applications. Biologically, enzymes are the foundation of all metabolic processes. Enzymes drive cellular respiration, DNA replication, and nutrient metabolism. Without them, these reactions would be too slow to sustain life. Focusing on lactase, its most direct application is solving lactose intolerance. Many people lack sufficient lactase in their digestive systems, so lactose from milk cannot be hydrolyzed, leading to bloating, diarrhea, or abdominal pain. Commercial lactase tablets, like the one we used in the experiment, provide exogenous lactase to help digest lactose, allowing these individuals to enjoy dairy products. In the food industry, lactase is added to dairy products to hydrolyze lactose into glucose and galactose. This not only makes the product suitable for lactose intolerant consumers, but also increases their sweetness naturally. Beyond lactase, enzymes have widespread uses. In pharmaceuticals, lilozyme acts as an antibacterial agent, and urokinase is used for thrombolysis. In industry, amylase is used in brewing and sugar production, while cellulase aids in biomass degradation. These applications highlight how enzyme research translates into practical solutions that improve our health and daily lives. Before the end of this video, I'd like to express my gratitude. Special thanks to the Bio 110 lecturers and lab instructors at Duke Kunshan University for their professional guidance. Their explanations helped me connect theoretical knowledge to experimental practice. I also want to thank my classmates for their collaboration during the experiment. Sharing ideas and troubleshooting together made the process smoother and more rewarding. Finally, and the most importantly, thank you all for watching this video.